Well, thank you. It's a great opportunity to be part of this FairCarboN research program. So I'm going to present GreenScale. GreenScale is for growing resilient ecosystems, enhancing nitrogen savings and carbon in agriculture, a leverage of efficiency. It's mostly about carbon and nitrogen in plant biomass. So why is it important to try to reduce our nitrogen inputs in agriculture? Well, because agriculture represents about 2% of the global CO2 emissions. It's about equal to the commercial flights. And nitrogen inputs also give rise to nitrous oxide emissions, and 60% of which come from fertilizers. And nitrous oxide is nearly 300 times the warming potential of CO2. And the question we are tackling in this project is how does the leaf chlorophyll content control the plant carbon and nitrogen fluxes in the plant biomass? And how does the chlorophyll content will alter the resilience of plants to drought, to elevated CO2 and to elevated temperature. So to design the project, it was a very ambitious undertaking in this axis 3 of the core. It was to try to be able, using a multi-scale approach, to integrate knowledge from the molecular level to the ecosystem level. And addressing a question at the plant level and at the level of the field. And the question we're going to address is the impact of chlorophyll on the agroecosystems. So we're going to talk about carbon and nitrogen in response to environmental stress, and we're going to use the different techniques we can employ to study these different processes at these different scales. And we've defined a very simple, with different work packages, one more at the molecular level, one more at the plant level, a field level, and the modeling at the larger scale of the ecosystems. And we have different people in different institutions on board. We have the modeling and data integration at the LSCE, CEA. We have the field trials with Arvelis as a partner. That's with Katia Beauchesne. I forgot to mention that's for Nicolas Richard is involved in this project as a partner. And the translation between molecular, plant, and field level it's a partner from INRAE Versailles, Fabien Chardon, the leader of the project, and myself. I'll be working more at the molecular and plant level. So chlorophyll is really a good molecule to study, to try to get this integration to work, because chlorophyll is a molecule. And it's embedded in large protein complexes in the chloroplast of plants. And you can observe chlorophyll with your own eyes in the field. And you can see the relation there is between chlorophyll accumulation and nitrogen fertilizers. On the left-hand side here, it's without nitrogen. On the right, it's with nitrogen. So there's strong interaction between these two. And of course, chlorophyll can be observed from satellites. So what's the bottleneck for modeling the leaf nitrogen partition? Here in this pie chart, that's the partitioning of nitrogen in the plant leaf. And about 1/4 of the pie is invested in chlorophyll and the reactions of electron transport, the bioenergetics of the leaf. And the second quarter of the pie chart, this one, is invested in rubisco, that is to say the carbon fixing reactions in the leaf. And these are very depending on each other because there's two modes of assimilation of CO2. There's a mode which is limited under low CO2 concentration. It's limited by Rubisco. Sorry, that's this one, the red one. Sorry about that. Limited by Rubisco. That's to say this quarter of the pie chart. And the other one is limited by electron transport. That is to say the other part. So what's interesting, and that's what goes in the land surface models, it's these set of data. You can see that across a large range of, a large variety of plant functional types, there's a linear relationship between the sorry, the pointer is not working very well. At the level of carboxylation and at the level of bioenergetics, and these two are strongly linearly correlated, and they depend on the nitrogen content in the leaves. The high nitrogen containing leaves showing a high flux, and the low nitrogen containing leaves showing a low flux. And that's exactly the question we want to address in the project. So to do that, we need tools and we need a model. We have chosen the C3 grass barley as a model. And this is a good model because it's the fourth most important cereal in the world after wheat, corn and rice. It's very much like wheat for the physiology of the crop. And it's diploid, so it's amenable to genetic analysis, for example. It has a natural variation, or the plant breeders have selected different varieties, some for molting and some for feeding animals. And the malt varieties are high in carbon, high in carbohydrate in the grain, while the varieties used for feed, animal feed, are high in proteins in the grain. And the working hypothesis we're placing is to use the natural variation there is in an enzyme, in a molecule, the chlorophyllide A oxygenase, and using a panel of induced varieties, non-GMO mutants, on this same gene, the chlorophyllide A oxygenase. And what do we expect in low chlorophyll crops? Well, they're less green, they contain less chlorophyll, so we expect less nitrogen assimilation from the soil. Under saturating light conditions, we expect a similar CO2 assimilation and remobilization of the carbon to the grain and exudation into the soil. And we expect also less evapotranspiration because these crops absorb less photons, less energy, so we expect the temperature of the canopy, the temperature of the field to be slightly lower and therefore have less transpiration. So possibly more resistant to drought. I'm afraid I don't have time for this, but I'd just like to show you that we have these green varieties. They're here in the lab. For the moment we haven't done any field trials, but it's in the project. They look very much different than the normal green cultivars. Sorry about that. Yes. So to put it very simply, we're going to measure the carbon and nitrogen pool sizes in the leaves and the fluxes of these components in the plant using labeling, using 13C or 19N. We can follow the absorption of CO2 through photosynthesis and the remobilization of carbon to the grain and exudation to the soil. And we can also measure very precisely the assimilation of nitrogen in the plant and then the remobilization at the stage of grain filling. And we'll do that at INRAE, here, there, and at CEA as well. And then we have all the previous expertise that is needed to understand, calculate, model the nitrogen assimilation and remobilization at the grain filling stage with previous studies that have been done and that we will do again for barley. Finally, we want to take these varieties to the field to see what having a low chlorophyll crop changes for biomass production in the field. And for this, we have two possibilities. The first one will be there at Gréoux-les-Bains in the south of France, next to CEA Cadarache. And the second one is going to be in the north of France, not so far from Versailles, at Ozuel-le-Marché, at this beautiful phenotyping facility of Arvelis. It's also interesting to have contrasted climatic conditions for the field trials, one in the south, one in the north. We'll compare in both conditions the high nitrogen content to the low nitrogen content in the fertilization, and in the south it's drier, so we'll compare the watered to non-watered conditions, and in the north we'll compare the normal rain-fed conditions to the rain exclusion conditions. As you can see here, they have a setup to block the rain. And finally, we'll go back to the parameters which are very important for the modeling at the global level. And from what we get as modifications or as insights from the experiments, we'll try to use as an input for the Orchidea model, which models the carbon and nitrogen cycle at the level of the land surface. How do we fit in the structure of FairCarboN? We certainly would like to have some interaction with other projects. We have identified, well, some people from the LSCE are involved in the Alamode project, and would like to contribute data that could be used jointly with this funded project and the targeted project Alamode. And we've also identified that the project Canet, which was funded in Axis 3, has the same kind of topic on the carbon and nitrogen cycles, but more focused on the soil and on the microbiota of the soil. And we would like to have a bridge with this project. Finally, what about the outreach? Well, for us, it's a really great opportunity to be part of a larger research program, because we'd like to integrate our research effort into a more concerted action. We'd like to be part of a bigger or more inclusive initiative, and would also like to nurture or contribute to nurture a vibrant community of young researchers. That's not just wishful thinking. We've been involved in 2 events this year. One was the summer school, which was organized by Fabien Chardon at INRAE Versailles this year. It was about how plant science can contribute to tackle climate changes. And the second one was hosted at Cadarache, mainly because of Zini Johnson, who organized this, and the European-funded Capitalized project. And it's to write I have 10 seconds left. Translational photosynthesis and the prospects for carbon fixation in crop improvement workshop. And my last slide is to thank you very much and to tell you, you can follow us on LinkedIn. I've just posted the poster that Fabien has made and which is hanging in the posters room. Thank you very much.
Thank you.